Welcome back, everyone. So we're here now for our second keynote of today on design and science, strategies and research. Because in our quest to foster innovation and create impactful solutions, it is essential to integrate diverse disciplines such as design and science. So by examining the synergy between these fields, we can develop strategies that not only address contemporary challenges, but also pave the way for groundbreaking advancements. So how can the intersection of design and scientific research drive us to rethink our methodologies and outcomes? And what role do psychedelics play in enhancing our understanding and reconnecting with ourselves and the world around us? So these questions are here today, and hopefully Grace will talk about that a bit. And I'm thrilled to introduce Dr. Grace Blest Hopley. She's a leading figure in the field of psychedelic research. She earned her PhD in the neurochemical and neurofunctional effects of cannabinoids, after completing a master's in neuroscience at King's College London. As the director of research at the Heroic Hearts Project, she's dedicated to investigating the therapeutic potential of psychedelics, particularly for combat veterans with PTSD. Grace's extensive background includes her role as a chief scientific officer at NWPharma Tech, where she leads clinical trials focused on psychiatric and neurological disorders. Her work leverages neuroimaging techniques to explore the mental health benefits of cannabinoids and other plant medicines. And additionally, she's the founder of Hystelica, a community dedicated to understanding women's biology for safe and effective psychedelic use. Grace is committed to advancing psychedelic research and is evident in her ongoing effort to bridge scientific rigor with therapeutic applications, aiming to transform mental health outcomes through innovative approaches. We're honored to have Dr. Grace Blest Hopley with us today to share insights and expertise at the second event at the intersection of psychedelics and design. Grace, the floor is all yours.
Hi, Floris. Hi, everyone. Thank you so much. And what a wonderful intro to receive there. Not sure where I found the time to do it all. So I will just share my screen now so I can bring up the presentation for you all. If somebody can perhaps give me a nod to let me know that you can see it, okay?
Yes, all good.
Excellent. So design and science strategies in research. So when I was first asked to do this presentation, I was thinking a little, what is there that could possibly be a link between design and science? Surely these are things that operate in completely different silos from each other. Science, of course, being led, majoritively by sort of slow and meticulous work in the pursuit of truth, whereas design is a far, far broader consideration, which only really cares about what things look like, right? So how on earth could we bring the two? But the more I came to think about it and the more I discussed with people on this call, like Alex, how these things could be somewhat combined, the more we realized that not only are they very much related, but actually there's a lot of lessons that could be learned between the two. So today, I would like to take you through a little bit of that, talking about an introduction about what sort of the two aspects of science and design and their intersections could be. I'd like to, discuss with you what the fundamentals of scientific trial design is, how we have the challenges and complexities of data collection, particularly in psychedelic studies. What does good look like? What does bad look like? It's very easy to make sort of generalized statements, but how do we see things going wrong or going right in scientific trial design? And how can we use design to optimize the scientific trials that we are conducting? And then finally, how can design learn from science and how can science learn from design? And then we'll touch a little at the end around what the future of scientific trial designs could be, and maybe hopefully discuss a few ideas amongst ourselves, particularly as we bring this into the focus of what are scientific clinical trials using psychedelics. I'm sure I won't be the first one breaking the news to a number of people on this call, but there's a lot of very interesting complexities brought about by trying to study substances that have such profound and felt effects as psychedelics. So here we go. I won't go through all this again. You all heard who I just was. My name is Dr. Grace Blest Hopley. I'm a neuroscientist, and I am a researcher who's been working in the field of cannabis and psychedelics for quite some time now. And within that position, I currently hold 3 positions that I think are potentially useful for me to give you some oversight. So you can see the kind of problems that I am facing on an everyday basis in the companies that I'm working for. So, starting from the left with NWPharma Tech, we're a biotech company that is doing clinical trials for a drug product. So this is a drug product that looks not dissimilar from potentially many other drug products that you may have taken, like an anti-inflammatory medication or any other psychiatric medications. It's a pill. You take it, before food, after food, at a set time of day. It will be prescribed from a doctor, probably, in a little bottle or a blister pack, and look and feel a lot like other drugs. Now, the reason I'm saying that is so that you can understand when we're designing trials for this, we want to be looking, obviously, at how people are going to be using these drugs when they actually get prescribed. And so it's really important for you to kind of understand how the therapy will be applied in everyday life. These are very much clinical trials. Clinical trials are trials which are largely done under a much higher level of scrutiny than others. They are involving human participants, and they need to go past quite rigorous checks from various ethics bodies, and often further checks from particular healthcare bodies when you are using participants in those trials who actually have certain diseases and disorders that you would like to look at. Now, slightly contradicting myself in terms of all of the trials that I have to look at and sort of design and overview in my role at NWPharma Tech. It includes preclinical trials. So this is clues in the name, preclinical. So studies that are happening in animal populations or even cell line populations. Phase 1 studies. These are the first studies that are usually conducted with any drug product or potential drug product. These are first in human studies. They aim to take normally a very small number of healthy controls, give them the drug product, and see what happens. See how much of the drug product is absorbed, see what the effects were, see what the side effects were. Really start to understand, what does this molecule look like in the human body? After a successful phase 1 trial where you've understood, is it doing what we think it's going to do, and is it safe whilst doing that? One would then move on to a phase 2 trial. This is kind of a little like a proof of concept trial, where you would take your drug product or the therapy that you are looking to investigate, use it in a small population of patients who are exhibiting the symptoms or the disease profile that you want to potentially look at your therapy for. And you would conduct this trial maybe in between sort of 30 to 60 patients. Now, that is enough to give you a proof of concept indication. See if the drug is working, see if it is treating what you think it is going to treat within these patients, and also to give you a little bit of an idea about the effect size. How much of an effect are you seeing from your drug product? And the reason you might want to know that is because you then would need to conduct what we call a pivotal clinical trial, which is a phase 3 clinical trial. That is a trial large enough, often with hundreds, sometimes thousands of participants with the disorder you're looking to treat, who you have done a calculation based on the effect size in the phase 2 trial. You know that at that number, if your drug works, you will be able to pick it up with the statistical analysis in a way that will be accepted by the regulatory bodies that be, enough that they will see, yep, that's great. We agree with you, your drug works. We agree with you, your drug is safe. Now go off and do another phase 3 trial, because 2 is the minimum requirement for most drugs. And then with this full package of preclinical phase 1, phase 2, 2 phase 3s, we will consider licensing your drug or treatment. So that's what I very much do in the clinical trial world, but it's not really what I do in terms of most of my research, which comes down to psychedelics. So in the middle panel here, I've been the research director for Heroic Hearts Project for nearly 4 years now. And what we do at Heroic Hearts Project is primarily take military veterans who are very much in need and deserving of therapeutic relief in some manner from largely post-traumatic stress disorder and traumatic brain injuries and the likes. And so we take them off to residential retreat programs. Now, of course, it's extremely interesting what we are doing, and we would love to be able to capture data around it and understand it. And in order to do that, we are able to conduct what are called observational trials. So with a clinical trial, I am very much designing what my intervention is. I'm very much putting down, right, you're going to take the drug at this time of day. This is how much you're going to take. This is when I am going to provide these different therapies to you. Whereas with an observational trial, as a researcher within this, I am not designing any part of the therapy. This is purely a research project that sits alongside an activity that has already taken place. But with an observational study, you can still be pretty probing in what you are able to achieve. So alongside Heroic Hearts, we are able to undertake a number of quantitative analysis techniques. This largely involves before and after taking of behavioral survey data, so very much quantitative studies asking about symptomology for depression, for anxiety, for post-traumatic stress, for a number of other things that are interesting to us as researchers in this project. We also have cognitive tasks that we ask them to do both before and after attendance to the retreat. And we've also got some imaging data where I've gone myself down to retreat centers with EEG machines, and we're able to capture before somebody goes into a psychedelic ceremony and retreat program, and once they come out, what is the neuroimaging in there? And then something that is quite interesting that we have just started to look to do is what we call qualitative research. So quantitative, clues in the name, it's quantifiable, you can add it up. Qualitative is much more interesting, and particularly when we're looking at psychedelics and really how do we capture some kind of degree of understanding around these drugs, around these substances, around the experience of taking them. And qualitative research is an incredibly useful tool for that. So in qualitative research, instead of saying, on a scale of 1 to 10, how bad were your symptoms of X today? Qualitative research will ask somebody, how did it feel when you did Y or X for that matter? And finally, my own company, which I founded, is Hystelica. We are a research, advocacy, and educational company that is focused on the intersections between women, women's biology, and psychedelics. Now, when looking at women and research around women, I unfortunately regret to inform you there has been a huge lack of research actually done. What that means is that we're operating in a space where there's not an awful lot of previous research or research theories from which we can build our new clinical trials. So we are in a position of what I like to call sort of pilot studies, hypothesis finding, hypothesis forming, where we are conducting a lot of observational research. So we're not actually providing interventions to people ourselves, but we are observing them by asking them to fill out surveys around their own experiences, trying to collect this a body of data from the collective knowledge of females who have used psychedelic substances. Often these are retrospective, so we're asking people what happened when you did this thing in the past, whereas with Heroic Hearts, we ask them before and after. And we are also looking to do longitudinal, so we are following people along a journey. So for instance, we are due to launch a study for women who are microdosing for symptoms of the menopause. And in that study, we will ask them to fill out several different time points as we go along. So that's an overview of all the research that I'm doing. And let's get into understanding a little bit about the fundamentals of scientific trial design. As I alluded to a little bit in my previous section just now, when we conduct science in any way, what we're always looking to do is build on what has come before us. With science, it's not about walking up to the map and sticking a pin randomly in it. It's about building the next brick along the path to somewhere. You are continuously adding to the total sum of human knowledge by putting one brick on top of the next. Now, the reason that we do that is because we are always having to build from other kind of known knowns. And the way that we do that is via a number of these methods I have written here. So conducting literature reviews is extremely important. So looking back at everything that has been peer reviewed. Just to touch on peer review for a second. In science, the way that we disseminate the results that we get from clinical trials is most often through peer review. The requirement of peer review is that you write a manuscript up in a very set format, usually introduction, methods, result, discussion, conclusion. And in there, you give all of the overview of how you came to decide to do what you were going to do, how you did it, what you found, what you think it means, conclude, and that's that. That then goes out to other peers who are within your academic field. They will give their feedback, often ask for amendments and review of what you're doing before the editor and the journal will agree to publish your paper. Now, the reason for that is it means that you can't just go and come up with something and put it out there and it will be peer reviewed. It must go through these processes, and that holds up a standard in terms of is it just beyond yourself that other people agree with what you're saying, that other people agree that, yep, this is reasonable science, there's reasonable theory and reasonable methodology behind what this person has done, therefore we will allow it to be reviewed. So that allows us to then, when building scientific design, trial designs, go through the previous literature, understand the theories, understand the fundamental biology and underlying neuroscience in the case of a lot of the psychedelic work we do. You can then do things like conduct meta-analysis or systematic reviews. This can then help you to sort of develop more those hypotheses in more thorough manner. You then come on to looking for how you might want to replicate previous stuff that was done in previous trials. So looking really into the method section that they have used. Can we do something similar so that we're going to say, we know we're going to do something that is replicating this previous trial, but with another variable, for instance. So we're building on what is already known, giving us these incremental advancements. Evidence-based protocols, of course, from being based on previous work that has been conducted. We can then learn from these previous trials what was done, what was not done. And finally, I've put ethical considerations here because it's essential to prove to the ethical boards when doing scientific research, that what you're doing has some basis in sanity. You're not just coming up and saying, I would like to do a clinical trial, we're going to do X, Y, Z. I have no real basis for why I think it's going to work. Because you need to convince an ethics committee that what you're doing, particularly when you are using human participants, particularly when you're using substances that can be quite powerful and have quite considerable effects on the body, you need to know that the reason you're doing it is sound. Now, every scientific trial starts with a question. And what is the question? So the question is what it is you are trying to answer. And it is the guide to everything in a study. The study should always start with the question. That's how you determine what your general objectives for it will be. It's how you determine what your methodology will be. And it's certainly how you determine how you will analyze the study. It should always address a specific gap in the current knowledge. So you will obviously understand that gap having done a literature review or a systematic review of all of the previous work that has taken place. And it has to be something that, of course, is testable and measurable. Unfortunately, I still even find myself coming to build a question, coming up with something wonderful, and then realizing we don't actually have any way of measuring this. And it's particularly interesting when we think about psychedelic trials. I'm sure many of you here have experience of psychedelics and what they can feel and look like. Now, trying to measure that is extremely difficult, and trying to do that in a way that is rigorous and answers the question is particularly hard. So, by asking the right question or defining the right question, we can then move on to what we are going to need to be able to answer that question. So one of the first things that we would have would be to develop a hypothesis. Now, a hypothesis is your idea of what is going to happen. So you say, my question is X, and I'm going to hypothesize that when something happens, this will be my outcome. It's very important to do that because it also defines part of the rest of the trial design, like what is the type of study we would need to do. As I said to you earlier, we can have, clinical studies, we can have observational studies, we can have different types of studies within them. We would obviously need to know what our sample size is, how many people are we going to include in this trial? What is the minimum number of people that we will need to be able to see a signal that what we are doing is having some effect? Now, of course, the population selected is going to be extremely vital in certain studies, but maybe not in others. Sometimes you're going to think about population selection and say, okay, well, of course, what I'm going to need is a population of people who all fit very, very specifically within this particular indication, this particular disease group. But other times we might say, well, actually, we want to see what is the general effect of psychedelics across the population, in which case you're going to want to try and get as much diversity into that group as possible. And how are you going to do that? How are you going to recruit those people? Do you need a control group? Do you need to have somebody who is perhaps taking a placebo, so a dummy drug? Or maybe it's just it's a group who is going to be of a completely different population from the population that you are targeting. Or it could just be minor tweaks, they're taking an active drug, but you want to understand what is the effect of a therapy component alongside a drug, in which case your control group would be without the therapy. Now, randomization, we will come to talk a little bit more about randomization later. Randomization is where we basically ensure that if we are going to have a number of different groups within the study, we are not cherry picking who goes where. Unfortunately, you do see a lot of this biases that get put in position, particularly if you get people recruiting in, for instance, from a GP surgery. And a GP has been asked, okay, well, we'd like to bring people into this trial, and there isn't a good, this wouldn't really happen with a good clinical trial these days, but definitely happened in the past, where they said, we want to try a new treatment. And the GP general practitioner is able to say whom goes into which group. Now, in doing that, it may be that they choose to put the patients that they know are very engaging, are definitely going to come back and do all the paperwork and stick to the trial design into the treatment group and not others. And that would, of course, bias your results, because potentially there is some psychological component to those sorts of people that mean that they would be more optimistic for the drug to work, for instance. And that's obviously just an example. Data collection methods. I went through a few of these earlier. Again, what data do we need in order to answer our question? Statistical plan, which is how we are going to analyze that data to answer our question in order to prove our hypothesis right or not. And finally, of course, timeline and resources, which I'm sure is not unique to scientific trial design and exists definitely in the design world also. It's all very well making the most beautiful clinical trial in the world, but if it's going to take you 3 years and cost you 10 million quid and you've got 6 months and £100,000, then you might need to think again. So, psychedelic trial design, complexities and the novel challenges that we face here. Now, a lot of the reason that psychedelic trial design is so difficult is because we really have had a huge gap in our knowledge placed from the lack of research that was done up until quite recently. We are definitely making huge advancements and definitely getting a lot more of a knowledge base up to be able to understand these substances more. But now we are, as I say, we've gone through that preclinical, we've gone through that phase 1, we've started to do a lot more of the phase 2, and people moving into the phase 3, and that is where we're really starting to run into the problems that we have, which is a lot around the subjectivity of the experiences, cultural and contextual influences that are around psychedelics and how we take them, how we integrate those experiences and the results from our trial. As many of you well know, the therapy doesn't stop at the 6 hours that you're sitting in a clinical trial space or even in a ceremony space. What goes on in the weeks and months after you've taken a psychedelic can be hugely influential on your overall outcomes. So how do we really get to the bottom of that? How do we balance our participants' safety with the scientific objectivity? I think is an extremely interesting part of this. Obviously, we have a lot of more understanding of what risk management looks like and what risk management needs to look like in a therapeutic sense. How do we take proper informed consent of participants when they've never taken a psychedelic before, perhaps never had an experience that allows them to go into this altered consciousness state? How do we really describe what's going to happen without biasing their experience of what is going to happen, but still say that we have taken informed consent? How do we, of course, offer that therapeutic support? And I guess part of this comes into the ethical oversight to participants in these sorts of clinical trials, particularly, as I say, if we're really just trying to drill down on what does this drug do? Well, how do we say, well, we need to look after these people when they're under the influence of this particular drug, but we don't want to look after them too much because we don't want to influence their experience enough that we might change the outcome of the trial. And then, I realized I have not changed the top thing on this. This is supposed to say something else. But one of the most difficult things that we have here is blinding, and that's blinding across both the participant and the researcher. I'm sure many of you have seen, there's a fantastic meme, I should have put it on this slide, of some people sitting around watching a group of people all dancing, saying, well, I guess we got the placebo. And when you are a participant taking part in these trials, often what we ask them at the end is, so do you think that you got the active or the placebo? In psychedelic trials, it's a very, very high success rate on people guessing which group they were in. And furthermore, under maintaining objectivity, it's also reasonably obvious to the researchers and the clinical staff who are alongside you. And then they can really change the way that they perhaps are interacting with you. We all are very, very very open to suggestion in our subconscious minds. And this is definitely true of researchers and clinicians as well, who are conducting these clinical trials. So what does bad look like? What does good look like in clinical trial design? I definitely know what bad looks like. We certainly see it, as I say, selection bias, which I alluded to a little before. I think selection bias is something that we've definitely seen happening in a lot of our earlier clinical trials. It's definitely happening an awful lot in the observational trials because of people having to self-select to enroll in such kind of research. It's often going to be people who have gotten a benefit out of it and want to share their experiences often. And that can mean that we miss some really, really vital data. So how we might get over that is, trying to sample people in more of a random way, trying to make sure that our recruitment and our outreach is such that we really make it clear we're not just looking for people who've had the best time. We really are looking for people who've had any kind of time from taking psychedelics. Inconsistent data collection, of course. I definitely have seen this happening a lot, where you have really long, boring, as to the next point, overly lengthy or complex studies. Then the participants don't really understand when they are supposed to be doing the next part of it, or they just get bored halfway through, turn the survey off. Why not? It's really, really boring. Another thing that will help you get inconsistent data collection is having repetitive or redundant questions. This can be quite confusing to participants. I know in some of the earlier studies that I designed for heroic hearts, I used to get these emails saying, are you sure? I'm sure I've already answered this question. And I just thought, oh, never mind. I'm sure people don't mind. But I started to realize that it was really affecting how many people I was maintaining to come back and fill in the next survey and the next survey. I'm sure you kind of realize that when you are designing anything, you need to hook people's attention, but you also need to be able to maintain it and not do something that is going to turn them off. Having unclear or ambiguous questions certainly does that. Nobody likes to feel unsure. And particularly when you're doing clinical trials, we're very much asking people to read these big information sheets before, put their signature and consent to something that can feel quite official and quite serious. And if you are then feeling like you don't fully understand it, you're not doing it right because the questions are not clear, well, that can make participants feel quite uncomfortable. Another thing that will make patients feel uncomfortable is overly intrusive or personal questions. Quite often when we're doing psychedelic trials, we are asking things that can be reasonably sensitive. Often these trials are around people having mental health difficulties that are based in traumatic experiences that they have had. If you've had a traumatic experience, how comfortable are you going to be just sharing that on a random survey with somebody? Probably not very. So you need to really think when you're designing clinical trials and you're designing the kind of questions, is this really necessary for me to know this level of detail about this participant? And I really think, you should try and collect as much data as you can, but be respectful around those kind of insights. Of course, you need to make sure that your all of your data is very well protected, and you're not going to have any data breaches in that sense. But also thinking about in terms of what the impact of asking somebody those sorts of questions could be. If you have a lack of incentives or compensation for participants, that's another big kind of turn off. People are normally very happy to assist and help with one or two quick surveys, but if you're asking for any great amount of input from them, if you're asking them to fill out numerous surveys time after time, it's always a very good idea to give them an incentive and compensation. But another point on this is to then not give them too much of an incentive or too much compensation, because if it becomes a lucrative aspect to be involved in your trial, then people may just take place for the incentive and the compensation, whereas you need to strike a happy medium. They're happy to give you their experience, their time, their data, because they are genuine participants that you want to speak to, but they will take some incentives and compensation. So complex procedures and instructions, this can be very off-putting. I do this all the time, and my colleagues in academia are absolutely awful for this and the other one here with insufficient communications, I suppose, and being monotonous and unengaging. We live in this academic world where we sit through these presentations where people present reference after reference, statistic after statistic, thick, dense text about things that we've spent years and years learning. And then we go and we design these clinical trials and we put these instructions in, like everybody taking this trial has also got a PhD in neuroscience. And normally that's not the case. And by doing such a thing, you are insulting your participants and turning them off from completing your study and completing your study correctly. Inflexibility in scheduling. Again, we always want as scientists, all of our participants to live in some kind of petri dish where we can control everything about them. And that isn't the case. A lot of the work we're doing, particularly in the psychedelic world and particularly with the observational work that I do, I am asking people to do this in their daily lives. I have to accept I'm not going to be able to get them to all sit down at their computer exactly this many hours before they do X activity and give me the answer. So letting people know that there is going to be some flexibility around what they can do in terms of the trial. And I think I covered off on the other two in my previous discussion. So what does good look like? I think I probably covered a lot of this previously as well, but concise and manageable study length. How much time are you really going to ask people to sit and give you? How much time are you really expecting people to sit down and concentrate? Unfortunately, the average sort of attention span of most adults today, thanks to the joy of the instant sort of dopamine hits we get through social media and the rest, is pretty short. I personally, unless you're going to be compensating people with something reasonably reasonable, and if you're going to be recruiting them quite specifically, if you're going to be conducting an online survey, you really don't want to be taking up more than 15, 20 minutes of people's time, making sure that all of your questions are clear and purposeful. One thing I like to do now is I send my surveys out to friends and family. I say, please, can you just go through that and just tell me, can you actually understand what I'm asking? Can you actually understand what you're supposed to do here? Respectful and justified data collection. I touched on this previously, making sure that you make it really clear why people are answering the questions that you are asking them. People are often very happy to share, but they need to know that what you're asking is for a reason and is going to be used in a respectful way, that they're make it very clear if any of their data will be identifiable in any way. Simple and clear procedures, flexible scheduling, as I mentioned, effective communication, and an engaging and varied study design. And maybe this is where we start to bring the synergies in between science and design, because it isn't just about asking questions. There does need to be some kind of aesthetic level to how we present this data. And it's definitely one of the ways that I would like to see more designers coming in and collaborating with scientists and researchers so that we can make our make our techniques, understand the questions we want to ask, understand how we want to ask them, but then bring in a design aspect to it so we can just make them look nicer, feel nicer, be quicker to understand, quicker to complete for our participants. So, how can we use design to optimize scientific trials? Think about the participants. We really need to identify our target demographic, understand where we're going to find them, access their capacity and willingness to complete the study, and provide appropriate incentives to ensure high participation and data quality. So, with that, I'm going to call on my work with Hystelica, for instance. I know that my target demographic for that is pretty much all, in all of the trials so far anyway, is women. In certain trials, it's going to be women of a certain age with a certain hormonal profile. And I really want to be thinking about that when I start designing my trial, start designing the adverts that I'm going to put out there. Am I thinking about something that is going to grab the attention and be aesthetically pleasing to my target audience, rather than just something that's very plain, very generic, and is less likely to capture their attention? I'm also going to think about where are the places where these, women who are experiencing perimenopausal and menopausal symptoms, where are these women hanging out? And we're not just talking about in the physical world, but very much now in the digital world. How can we go into those spaces where we know our participants are going to be, and go in there with really clear and concise information about our trial, but also designed in a way that's going to grab them and bring them on board. So that advert that I've made, that I'm going to be pushing out in the next week or so for my study in menopause, looks an awful lot different from the advert I'm putting together that's going to go out for my clinical trial that is looking at people who are clinically high risk of psychosis and most likely to be young adults, largely men, between the ages of 16 and 25. It's going to be a whole different color palette, font, the lot of it that is going to capture their attention. What is the patient burden? So this is, the total burden that is required of the participant in order for them to enroll and complete my trial. We have to evaluate what those demands are. What is the time commitment? How complex is the task? If it's an extremely complex task, am I really going to get them to read, a whole page or 2 pages of text? Or would it be better if I made a video that they can just watch within 10 minutes, and then they will fully understand the trial? How many visits am I going to need them to do? Can I bring that down? Can I potentially roll visits into one so that I'm collecting bloods on the same day that I'm doing the cognitive test, so that we can cut that in half? Potential physical and emotional strain. It's very likely, as I say, with psychedelic trials, we are trying to assess often, how these things have been affecting people in terms of trauma. Well, then I need to know that I have got the right resources that I can signpost people to should they need it. So often we are required by ethical committees to be able to provide access to certain therapists and counsellors alongside the study, and ensuring that the study is manageable and does not deter participation. Furthermore, how easy are the questions to understand? We need to ensure that the questions are clear, straightforward, free of jargon, minimizing the risk of misunderstanding, ambiguity, or misinterpretation, so that participants can easily comprehend and accurately respond without confusion. And finally, how easy is the study form to complete? The number of study forms I've seen that are just so long and boring and not clear, and you can miss out questions, leading to incomplete data collection because perhaps somebody has designed it and the last question falls off and goes on the other page, and then nobody answers it. And then you can't get the full total number required for that particular survey because one question went on the other side of the page. Making sure that it's user-friendly, well-organized, clear instructions, simple language, logical flow, ensuring accessibility across all devices. It's very, very rare now that someone's going to hand you a piece of paper and ask you to fill it in. What does, this look like when somebody tries to do it from an iPhone? Is that going to be possible for them? So, how can design learn from science, and how can science learn from design? So design could definitely learn from science, I believe, in a few different ways. I think that evidence-based decision-making is a really great handrail for science. It's very, very rarely, as I say, that you get any kind of real, random, brand new dot in the map that you need to come out from. You're always building from this existing knowledge out there and relying on data that is already there. And when we are working in a design field, often it can be difficult starting with a blank piece of paper. And so looking for previous work, looking at previous data from previous design of what was effective and what was not, can then help you to then say, well, I'm going to do that, but add my own novelty, add my own aspect, or bring in this new component to it. We also have in scientific research systematic evaluation and testing of our work. This can give us really good feedback of whether something is working or not. It can allow us to, make these small tweaks to our hypothesis, to our theories, until we get to we never get to the truth, but we can get closer to what we know to be true. And in design, this could be very much applied. And I'm sure many of you are also involved in that kind of work with your design, where you might make something and send it out for feedback from, I've forgotten the name now, not control groups, from opinion groups who are perhaps representative of who your population you're trying to reach are, and then use that as feedback, which you can tweak. And of course, that is a form of measuring the responses and the outcomes. And you can do that both with quantitative metrics, out of 0 to 10, how much do you like the font on this? How much do you like the color? How much do you like this, that, the other? And give yourself very much quantitative metric where you could then sum up from a group of 100 and get some sort of really very clear numbers that would quite quickly show you, or the qualitative feedback, going and interviewing people, asking them, how, what's the first thing you thought about when you look at this particular design? Now, how can science learn from design? Definitely a lot of interesting things that, particularly since having my conversation with Alex, I've been able to bring in here. So for design research, we can certainly start thinking a lot more about user-centered approaches. With design, the user really does sit a lot at the heart of what is being produced. Whereas in science, you would be surprised how late the participant in the trial actually comes to be the focus of the researcher's mind. This enables for if scientific design was to use this more, we would be able to have more empathetic understanding and inclusive designs to our clinical and observational designs. Storytelling and a narrative. People love stories, I am just finding out. By having compelling narratives and contextual framework for the studies that we put together, we can kind of bring participants in, get their buy-in a lot more. They can understand what we're trying to do. And when people understand what you're trying to do, they can get behind it, and they're more willing to put in a bit more effort. They're more willing to come and do the next survey and the next survey, because you have effectively told them why it is important that you want them to be involved in this. And of course, innovation and creativity. We don't always need to be just following exactly by the rules, how it was done before, how the person before us did it. By actually allowing a little bit more innovation and creativity, which we certainly are seeing and needing in psychedelic trial design, we're able to do a lot more creative problem solving around things like blinding issues that would perhaps are not been so clear in what is the very rigid and old way of doing scientific design. And it gives us the opportunity for much more flexible approaches. So the future of scientific trial ideas, particularly looking at psychedelic research, I think we need to come up with much better strategies for mitigating the bias in psychedelic research. That certainly is a big issue with obviously blinding, and blinding for both the participant and the clinical researcher involved. Having that importance then of objectivity and impartiality brought in to our psychedelic trials by having better blinding. Having better considerations for the diverse patient groups in psychedelic studies, we certainly have had an issue with that. We had a lot of our original trials. Oh, sorry, that's my dog. A lot of our original trials required us, because of the huge effects that we can have from psychedelics, to use a lot of participants who already had experience with psychedelics. Now, what that did is it meant that you really, really cherry-picked a very subset of the population who were often, I hate to say it, sort of white, middle class, living in metropolitan cities close to the big universities, who were doing the research. And they were not very representative of your average Joe on the street, who is probably more likely to be the person who ends up getting a prescription for whatever the indication that the trial is going forward to. So we definitely need to ensure that we have inclusivity and representation in our research samples, so that when we do get to that big phase 3 part of, does our drug work in a big enough population that we can say without reasonable doubt that this drug works? Well, can we say that in a population that is representative of the population that the therapy is intended for? So apparently, this is something that I said on the Psychedelic Design podcast, that in psychedelic research, we have to honor the profound personal transformation experience throughout the journey, from preparation to integration. As scientists, we must respect individuals' processes, guided by ethical bodies and understanding the sacredness of the full journey. Now, the reason I said that is because we are really operating in psychedelic science in a very interesting place. And when I look at the work that I do with Heroic Hearts and with Hystelica, I am trying to kind of go around and measure something that is so personal and fundamental to the person who took the psychedelic, who went to the retreat, who took part in the ceremony, that it would be wrong for me to interfere with that in any way. Now, when we extrapolate that out into the clinical trial space, it's a lot more difficult, because obviously we do need to go via certain sort of FDA guidelines, MHRA guidelines, for instance, in order to collect the quality data that is required. But I think it's really, really important, once we've thought about the blinding, once we've thought about the population, that we also think about the individual and how transformative and important that individual experience is. And as researchers, we should be careful not to interfere and get in the way of that particular process. So, thank you very much. I have rambled on for a little bit longer than I was perhaps thinking, but I would love to first thank you all for listening, but secondly, ask if there are any questions that we may have, or anything else anyone would like to ask about the presentation I just gave, or any of the research that I am currently undertaking. So thank you.
Yeah, thank you so much, Grace. It was a great presentation and super interesting to learn about. There's different perspectives from the clinical research to the observational, and yeah, also how you're thinking about design, how those worlds interact. I have one question. We'll do one question here live, because otherwise we'll still have a meeting after this, or a presentation after this. And this question was asked a bit earlier during your talk, a half hour ago. So maybe you've answered this, or think you might have answered this already, but maybe you can riff on this. Do you have thoughts on lessons learned for future trial design coming from some of the issues that have come out of the trials that are currently further along in the process?
Yeah, $1 million question. Yeah, I do. I think that particularly, I'm assuming that we are referring to the MAPS trials and the MAPS data that is in phase 3. We also have Compass Pathways, of course, who've got some phase 3 work coming along. And I think that we need to really look, like you say, at what the design of those studies were, and just ensuring that the protocols themselves were conducted in the most rigorous way possible, because when you are a first mover in this space, people are always going to be looking for any sort of minor errors or mistakes that are within your design. So for those trials, they needed to be absolutely watertight and unbelievably rigorous in their design, because they were very much first movers. Now, when it comes to the blinding situation and what we do about that, I mean, it's a bit of $1 million question. And I actually think, as a neuroscientist and understanding the way that psychedelics work, it's almost impossible to remove that kind of placebo effect, because in a way, placebo itself is a change in your subconscious perspective of what is happening, and that's sort of part of the mechanism of action of how psychedelics work. So they change some of your subconscious perspective of how things are, and your belief system around certain things. So I would like to see the way that we look at, those first trials have happened. Obviously, we can't go back in time and change them, but people looking to collect not only that research in phase 3 trials, where we would have a control group and an active group, but looking at ways that you could perhaps remove, just the very part that you are looking to interrogate further. So, maybe it's a high dose of MDMA and you could use a smaller dose of MDMA. There are some trials that look for other drugs that bring about similar kind of physiological effects, although are not having the same neurological impact. Although the ethics around that, I think, is a little dubious. But for me, what I think is going to be the most important, what I think is going to be the thing that we should start looking at, and I hope that people like the FDA and the MHRA and the EMA accept, is looking at much larger bodies of open label studies, where we all know everyone is going to get the treatment in it. And by using much larger studies, perhaps in a phase 4 design, which I did not include in my presentation. That's a whole presentation for another day. So post-market authorization, every person who is then prescribed the therapeutic is required in some way to be monitored in the same way as in a clinical trial, so that we're able to build up, okay, we don't have a placebo control comparison in 300 people, but we have the data from 3,000 people who took the drug. And from that kind of data, we can then do other types of statistical analysis that I believe still give us very rigorous evidence that what we are doing is effective. And then we can also look more within that to do things which I am always shouting a lot about at Hystelica, looking at things like, well, are we then seeing variability within the subjects? Are women responding the same as men? Are women in the estrogen phase responding the same as women in high progesterone phase? These kind of questions. So we then get to understand the drug a lot more in its kind of real world context.
Yeah, Grace, thank you so much for your answer, for your presentation. It's great to learn about what you're doing, and as you said in the beginning, like how do you have the time to do everything? It's so great to see you contributing to the space.